Hey guys, welcome to Rocky by Chem series. Today's session is going to focus on DNA library construction. Please, if you have not subscribed to my channel yet, kindly support me by doing so. I appreciate all your help and support. A library is known to be a place whereby books are stored. So when you visit any library, you will come across different books like biology textbook, molecular biology, microbiology, engineering textbooks, mathematics, and then etc. And each of the books have specific information within them. Therefore, a DNA library literally means that a library containing DNA. When you consider living organisms like human beings, the DNA have different genes. For human beings, we have about 20,000 to 25,000 genes that are found in our entire genome. And each of these genes code for a particular mRNA and then protein necessary for a particular function. So all the various genes here can be used to form clones and then stored as a library. So this is what a DNA library means. You have your clones, and each of the clones represent a particular fragment of DNA. That is what a DNA library means. So as I move forward, I am going to show how DNA library is constructed. Now, as I move forward, I will be making mention of the word clone. So in case this is your first time of meeting the term clone in molecular biology, a clone is just made up of a vector and then an insert, a vector and then a fragment of DNA. So when you insert your DNA fragment into a vector, then you will obtain a clone. A vector in molecular biology, to be precise, in molecular cloning, just represent a DNA, normally circular in structure, that we insert our DNA fragment into it to form the clone. And it is the clone that we will transform into an appropriate bacteria for multiple copies to be obtained. So this is what a clone means. A clone is formed from the insertion of your DNA fragment into your vector. Now, let me move on to talk about the various types of DNA library. We have two main types of DNA library, and then they are genomic DNA library, that is gDNA library, and then complementary DNA library, that is cDNA library. So, genomic DNA library, as the name implies, is made up of the entire DNA fragment obtained from the genome, whereas complementary DNA library or cDNA library is made up of the various genes that are only expressed in a particular organism. So we target the mRNA, which represent the genes that are expressed, and then use the mRNA to form complementary DNA, and then use the complementary DNA to form the library. I will do another video to explain how cDNA library is constructed, so stay tuned for that video. The focus of this particular video is on genomic DNA library construction. So, as I said earlier, a genomic DNA library is a collection of clones representing all fragments of genomic DNA in a particular organism. So since our focus is on all fragments of genomic DNA in a particular organism, then gDNA library is constructed for only prokaryotes. gDNA library is only suitable for prokaryotic genome. And the reason is that prokaryotic genome have no introns, and they also have relatively smaller genome size. When you consider eukaryotic genome, to be precise, human genome, it is made up of a chunk of intronic sequences. So it doesn't make sense to use some sequences that do not code for a particular protein to construct the library. It doesn't make sense. So for such eukaryotic genomes, we use cDNA library. We rather construct cDNA library for eukaryotic genomes. But for prokaryotic genomes, we focus on gDNA library. And another reason why gDNA library is constructed for prokaryotes is that they have relatively smaller genome size as compared to eukaryotic organisms like human beings. So when you consider human beings, for instance, our genome is made up of about 3.8 billion base pairs, and constructing a library for such an entire larger genome, it will be very difficult task to do. That is why the focus for gene DNA library construction is on only prokaryotes like bacteria. So the various steps in gene DNA library construction include the extraction and then digestion of DNA from the prokaryotic organism. So the first step is to extract your DNA from the prokaryotic organism and then digest the DNA into smaller fragments. The digestion is done by using a particular class of enzymes called restriction enzymes. These class of enzymes are special because they have the ability to recognize some specific sequences in the genome and then cut those regions into smaller fragments. So after obtaining the chunk or like the very large genomic DNA from the organism, then we will use the restriction enzymes to cut them and then break them into smaller fragments so that we can use the smaller fragments to create clones. Now, the second step involves cloning of your smaller DNA fragments into an appropriate vector. So when I started, I made mention of vector as a fragment, a particular DNA, normally circular in shape, that we will clone our DNA fragment into it to form our clones. So we have to, first of all, digest the vector. We have to break the vector by cutting it to open it. So restriction enzymes will be used to recognize some specific sequences in the vector and then cut it at those regions to open it. So when you consider this restriction enzyme, for instance, BBS1, it can recognize the sequence like CAA sequences in the vector like the plasmid and then snip at those regions to cut it and then open it. We have different examples of vectors. We can have plasmid, we have cosmid, we have phosmid, we have BAC, that is bacteria artificial chromosome, we have YAC, yeast artificial chromosomes, and a lot more other vectors available. And their selection is based on some choices like the size of the genome of the organism that you are working on. You can't use a vector like cosmid or phosmid for a genome that is very large. You can't use such plasmid for larger genome size. But when you are working with a very larger genome, you can go ahead and then use bacteria artificial chromosome. That particular vector will really work well. But when your genome size ranges from either 19 kilobase pair all the way to 23 kilobase pair, you can go ahead and then use cosmid or phosmid for your cloning. So after cutting and then opening the vector, then we will add the DNA fragment that we first of all digested from the first step. We will add it to the digested vector to form our clone. So we will insert the smaller fragment into the digested vector and then we will obtain our clone. And now, after obtaining the clone, the third step involves the transformation of the clones into a bacteria. We have to transform the clones into a bacteria like E. coli for the E. coli to replicate and then divide for us to obtain multiple copies of the particular clone within them. This is how the library is basically constructed. So we extract the DNA from the organism, we digest it, and then we will digest our vector as well, and then we will clone our DNA fragments into the vector and then transform it into a bacteria for the bacteria to replicate and then divide to obtain multiple copies of our clones. Transformation just involves the process of introducing the clones into the bacteria. This is what transformation means. So I hope this video helps, and kindly stay tuned for my next video on cDNA library construction.